So good morning all of you. I welcome you all for BioS 110 lab. That's the biology of cells and organisms. So today we will be learning something about the different forms of CO2 in the cellular and the pulmonary respiration. Overall, the view for today's lab is we start with defining the objective of this lab. Then we go ahead with the mini presentation where I'll delve deeper into the different forms of CO2. And then we have an experiment which defines the different roles of CO2 in cellular respiration. So let's get started with defining the objective. So the objective for today's lab is to understand the different forms of carbon dioxide in the blood and how they are ultimately removed from the body. So as you all know, CO2 is a very notorious molecule which has been a major problem and causing the global warming. But we also need to understand that CO2 is an important molecule that plays a very crucial role in our body. So when CO2 comes in contact with water, it forms carbonic acid, and then the carbonic acid is relatively unstable and it forms the bicarbonate or quickly dissociates into bicarbonate and a proton. So let's go into the cellular respiration. So when the cell undergoes a metabolism, so it basically breaks down the glucose into forming ATP molecules or deriving energy out of it, we have the CO2 which is end product as a byproduct of the reaction. Now the CO2 has to be removed out of the body because it's of no use to the cell. So then when the CO2 is now exhaled out of the cell, the red blood cells come into contact and quickly pick up the CO2. The water present inside the red blood cell quickly converts the CO2 into carbonic acid, and as I said earlier, the carbonic acid is relatively unstable and it quickly then dissociates to formation of a bicarbonate and a proton. Now the proton that has been formed, the hemoglobin molecule, then takes care or tries to stabilize that proton itself. Now once this is done, or once the CO2 is now converted into bicarbonate, the same red blood cell then can move to the pulmonary respiration cycle, where the red blood cell is near to the lungs, and then the bicarbonate itself then gets reversed to formation of this carbonic acid, and then the carbonic acid again comes back to the CO2 and it then exhales into the lungs, and in this way as we all know that we inhale the CO2 and we exhale the CO2. So in this way, now we can understand how the cells then produce the CO2 and how this CO2 is then quickly removed from the cells and then exhaled by the body, right? So in an experimental setup, so the way to understand if the CO2 is then converted into bicarbonate or not is by using a phenolphthalein indicator. So the purpose of this indicator is to turn or change the color when there is a change in the pH. So when we start with just using a pink colored solution which has a pH of roughly 8 to 10, when we continuously bubble the CO2 inside this pink colored solution, because the CO2 comes in contact with water, it forms carbonic acid, and the carbonic acid dissociates into bicarbonate and the proton. So the proton molecule or the proton itself is responsible for changing the pH of the solution. And that's why after some time you would see the change in the color of the solution. So which brings us to the next activity for this lab. So this is the activity where we have 3 modes of experiments, where we have the first one is the resting experiment, where you will just quickly note down the heart rate, blood oxygen level or the percentage, and finally you just note down the time required to neutralize the phenolphthalein solution, or the time required that for changing the solution from pink color to the colorless solution. And the rest of the 2 experiments would be just like after you continue your after you have your exercise. You can do a 1-minute exercise, you can do either a jumping jack, or you can just simply jog on the same place or same location to just simply increase the heart rate. So I will just, for the demonstration purposes, I'll just start with the resting phase experiments. We already have the phenolphthalein solution with us, and I think we also have the oximeter with us. So here we go. This is the oximeter that we have in here, and we have the respiration chamber with us. So to note down the time required for this, for the changing the solution from pink color to the colorless solution, we can use a stopwatch. So I have a stopwatch with me in here. So let me just quickly pull up the stopwatch mode and let's get started, right? And here we go. So currently my heart rate is like roughly 84 and blood oxygen is 77 or 97%. So let me just start the bubbling. Yeah, let's give one more minute. I guess that should be good enough. Yep, almost there. And here we go. As you can see, the solution has now turned into a colorless solution, and it's just because of the formation of bicarbonate and the protons, which has now changed the acidity of the solution, and the phenolphthalein has now turned into a colorless solution. So with this, I request all of you to get started with these 3 experiments. One of you can take lead on noting down the heart rate and the blood oxygen, and one of you can be the person who does the 1-minute exercise and the 2-minute exercise. So before we end with this, I just want to ask a simple question in here is, do you think that when you have a 1-minute exercise and a 2-minute exercise, do you think the time required to neutralize or time required to change the pink color to the colorless solution will increase or decrease? So with this question in mind, I request all of you to get started with this experiment. And as always, if you are stuck with anything, I'll be happy to help out with you. Thank you again.